Hello everyone and welcome to ASA level biology classes. Today we are going to have lecture 2 from chapter 1 cell structure. In today's class our learning objective will be to understand what is resolution and to know the working of electron microscope and its types. Let's understand what is resolution. Now if you look at this picture here, this is a picture which has been taken using light microscope. This is a plant sample, plant cell, and this plant cell has been observed under the light microscope. So this picture is called as photomicrograph. On the other hand, this picture is the same sample, same plant sample, but this sample has been observed under the electron microscope, so it is called as electron micrograph. It is called as electron micrograph. Now you can easily see the difference. In case of photomicrograph, you can hardly see any cell organelles except nucleus and cytoplasm and cell membrane. But if you see the electron microscope, you can observe different type of cell organelles. Why do we have such a difference in photomicrograph compared to electron micrograph? And the answer is resolution. Now let's understand what resolution is. Now the basic definition of resolution is the ability to distinguish between two objects very close together. The higher the resolution of the image, the greater the detail that can be seen. That means if you have two objects here, if there are 2 points here, and if these 2 points are observed as 2 points, not as a 1 point, then the resolution is better. Whereas if you see these 2 points as one single point like this, then of course we can say that the resolution is not that good. So as you're increasing the resolution, you will be able to see not one but 2 points or 2 objects. So in a cell, if you have different cell organelles, if the resolution is not good, if the resolution is not high, then you will not be able to see the cell organelles as you have seen in photomicrograph. But as you are observing the same sample under the electron microscope, which we will be discussing now, you will be able to see many different types of cell organelles inside. Now the amount of magnification, the magnification which we are doing, depends greatly on resolution of the microscope. If you are trying to magnify beyond the limit of the resolution of the microscope, then the image that we will see will be the blurred image. It is not same as if you have a photograph. If you have taken a photograph using a camera, and if you try to magnify it, you will be able to see certain details, but not in case of microscope. In microscope, if you are trying to magnify beyond the limit of resolution of the microscope, the image will just get blurred. Now why is it so? Why this resolution is so much creating problem in observing the samples? Now resolution, as I told you, linked with the nature of the light. So I'll just write down here, resolution greatly depends on the nature of light. Nature of light. Now when I'm saying nature of light, as we know, light, the property of light is that it travels in waves. So the length of the wave is basically, we call them wavelength. Okay, so there are various characteristics which we can give to the light, like for example the energy of the light, radiation, the wavelength of the light, okay, and frequency of the light, and there are much more. I'm not going much into detail in physics, but what we are really concerned here about the energy, wavelength and frequency. Okay, so if we are talking about the light microscope, in light microscope we are using light waves ranging from 400 nanometer to 700 nanometer. So in light microscope, in light microscope we are using light waves, visible light waves. We are using visible light waves, and these visible light waves has a range from 400 nanometer up to 700 nanometer. Now here, before I go further detail into this, I just want to tell you a very simple relation between the energy and wavelength. Now energy and wavelength has a relationship which is just reciprocal. They are reciprocal to each other. That means if a light wave has a very high wavelength, then the energy of that light will be very low. If the wavelength is very short, I mean the wavelength of the light is very short, then the energy of that light will be very high. So these two are anti of each other, energy and wavelength. Okay, so we'll just keep this in mind. And if you're just looking at here the resolution, so let's get back to the light microscope. As I was telling, that in light microscope the visible light, we are using visible light wave which has a range from 400 nanometer to 700 nanometer. Here we are talking about the wavelength. This is the wavelength. Now this range which we are using, if you see, the lowest wavelength that can be used using light microscope is 400 nanometer, and resolution, as I told you, depends on the nature of the light. So actually resolution is half of the wavelength that we are using to observe the sample. So half of the wavelength, half of the wavelength used to observe a particular sample in a microscope. So if we are talking about a light microscope, we are using visible light waves, and the minimum or the minimum wavelength, the lesser wavelength is 400 nanometer that can be used. So the half of 400 will be how much? If you try to calculate for the light microscope, it will be half of the wavelength. That means 400 nanometer, it will be 200. So 200 nanometer is the resolution of light microscope. So this is how we are calculating the resolution of the microscope. So it is, as I told you again, it is greatly depend on the nature of the light, especially the wavelength, and we know that higher the wavelength, lower will be the energy. Higher the energy, lower will be the wavelength. So if in a light microscope the lowest wavelength that can be used is 400 nanometer, and if you make a half of it, it will be 200 nanometer. So 200 nanometer is the resolution limit of light microscope. Similarly, we can calculate the resolution of different type of microscopes that is available, depending upon what is the minimum wavelength that it is using. Now, if you look at this particular picture here, this is basically the electromagnetic spectrum. Okay, this is basically electromagnetic spectrum, and this electromagnetic spectrum, as you can see, this 400 nanometer to 700 nanometer is basically the visible light. Okay, and this is the light radiations which we are using for observing specimen using light microscope. And again, I'm just repeating this, if we are using light microscope, we can have the resolution that is 200 nanometer. Why? Because the minimum light radiation wavelength that can be used using light microscope is 400 nanometer. Half of 400 is 200, so 200 is the resolution of light microscope. Now what if I want to increase the resolution? Simply I can simply go below 400 nanometer, because lesser the wavelength, lesser will be the lesser the wavelength, higher will be the resolution, as I understood, right? So if I want to increase my resolution, I can simply go below 400 nanometer. I can simply go for maybe I can go for radiations like, I can use radiations like UV, or I can use radiations like X-ray or maybe gamma rays, right? So that can be used in order to increase the resolution. But the problem is, if we are using such radiation, people have actually, scientists have started, they have tried using these radiations, these light waves, in order to observe the samples. But the problem was, these rays are very difficult to focus. So there's a difficulty of focusing, especially if you're using X-rays and UV, it will be difficult to focus. So instead of that, scientists, they have started using electrons. So this electron beams, or electron beams have started using in order to observe the samples, and hence because the electrons were used, we call them electron microscope. Now if I'll just go to the next slide, I just want to show you here, this is a picture which I got it from the book, Mary Jones biology course book. From there, if you see, this is the radiation as I was talking about, okay, and this has a wavelength of 400 nanometer. And if you see, this is a cell organelle called mitochondria, which has a diameter of 1000 nanometer, and you can see that this 1000 nanometer diameter mitochondria can be observed using this wavelength, because it is able to interfere within the wavelength, within the light waves, and hence it can be observed. Whereas if you see the second the other organelle, for example here ribosome, has a diameter only 25 nanometer, which is way less than the resolution limit of a light microscope, which is 200 nanometer. So resolution limit of light microscope, we know it is 200 nanometer, and 25 nanometer is way less compared to the 200 nanometer, which is a resolution limit of the light microscope. So it will never be observed using light microscope. So that is the reason we have to opt for some other light radiations which has a lower wavelength. So we need to opt for a light radiation which has a lower wavelength. But as I told you previously that if we are using wavelengths like X-rays and UV, it will be difficult to focus them. Hence electron beams have been widely used. Now why electron beams are used and why it was successful? Because electrons, as we know, electrons, if I'll just write down here, if electrons are basically negatively charged, we know that they are negatively charged, and these electrons, they are a part of an atom, we know that. And whenever we heat any metal, then what happens, these electrons, they just escape from their orbits wherever they are, and when they are escaping, these free electrons actually behaves like a electromagnetic radiation. So for example, if I'll show you, if this is a atom, okay, and this is a nucleus here. Of course, this nucleus is not the nucleus which we have it in the cell. This is the nucleus in the atom, which has protons and neutrons, and there above that there will be orbits where you have the electrons. Suppose this is one electron we have, and this is one metal. If we try to heat this metal, what happens is the electrons which are in the outer shell, that is valence electrons, will just eject out. And when these electrons eject out, they have very high energy and very low wavelength. So they basically behave like an electromagnetic radiation. Now, why these electrons which are acting as electromagnetic radiation are very successfully used in order to observe specimens? Because these wavelengths, as I told you, they are negatively charged. They can easily be focused using electromagnets. If you remember about the light microscope, we are using a lens in order to focus the light onto our sample, and onto the we are using objective lenses, we are using eyepiece lenses. But in case of electron microscope, because this is electron beams, we need a different kind of lens, and we call them electromagnets. So here the lens which we will be using will be the electromagnets. Okay, so these electromagnets will act like a lens, and they will focus the electron beam onto the sample, and so that we can observe. Now, this electron microscope has a resolution. Of course, the resolution will be very high. It has a resolution of 0.5 nanometer. That is the maximum resolution that can be achieved using electron microscope, which is actually 400 times better than the light microscope. Light microscope has only 200 nanometer. This is for light microscope. Whereas in case of electron microscope, the resolution is 0.5 nanometer. So you can see there is 400 times better resolution compared to light microscope. Now, the next thing is that we need to understand is about the types of types of electron microscope also, and the pathway of the electrons, which are how they are moving and how we can observe the samples. Now, if you see here in this picture, this is the source of electron, which is electron gun, an anode, which basically produces the beam of electrons. So this, what you see here, produces the beam of electron, and this beam of electron is going onto the condenser electromagnetic lens. Now remember, as I told you, here we are not having those glass lenses, but because we are using electron beams, we have a special lenses, we call them electromagnets. So this is a condenser electromagnet which directs all the electron beam onto the specimen over here. So this, what you see here, is the a condenser electromagnetic lens, and this, what you see here, is the specimen. So all the electron beam will go onto the specimen here. This is the specimen which is placed onto the grid, and you have an objective electromagnetic lens which produces an image. Then there will be a projector electromagnetic lens just below that, and which focuses and magnify image onto the screen. So you have these intermediary electromagnetic lenses, and then finally you have a screen which is a fluorescent screen in order to observe the image. Now why do we need a fluorescent sheet or a fluorescent screen to observe the image? As I told you, electron beams are not, we cannot see electron beam as in case of light microscope, because the light which we are using in light microscope was under under visible range, so we can observe through our eyes. But in case of light microscope we cannot, because we are using electrons. So we need to project these electron beams onto a screen, onto a fluorescent screen. And what happens here is, whenever the wherever the electrons hit the wherever the electrons hit onto this fluorescent screen, they basically create a black and white picture. So wherever the electron has been, suppose the electron has hit here or here or here, so you will see those areas become very bright, shiny, whereas the other areas will be dark. Okay, so that will give you a overall picture of the sample which you have it here on the grid. Now we can increase the contrast using certain heavy metals as a stains. Okay, so because these heavy metals which basically stops the pathway of electrons, and they create a better you will be able to see more densely stained paths, and using these metal atoms. Okay, so also some of the time scientists, they use false color images using computers. They use it in order to color a standard black and white picture which is being observed on a photographic plate. So they just make it colored so that we will be able to differentiate different organelles. Say suppose we are observing the cell and cell organelles, so we will be able to observe different cell organelles better if we color them. So that can be done using false color images. Remember one thing which is very important, that the entire electron microscope is actually in a vacuum. The reason is because these electron beams, if it is not in a vacuum, then what happens is there can be a chance that the air or the light might interfere with the electron beam, and we will not be able to focus these electron beams onto the samples. So it's important that the entire thing should run in a vacuum. Now because we are because this electron microscope is in the vacuum, so it is important that the samples which we are using, we are completely dehydrating it. The reason is, at vacuum, even at a room temperature, the water can boil. So if your sample has any amount of water in it, then it might damage the entire apparatus. So it is important that we must dehydrate the entire sample. Once the dehydration has been done, then we can use the sample to observe it under the electron microscope. Now let's understand the two different types of electron microscope, that is transmission electron microscope and scanning electron microscope. Now in transmission, if we are talking about the scanning electron microscope, if you see here, the main idea is same, that is we are using the electron beam only, but how we are observing it is different, and what we are observing it. Now in scanning electron microscope, the electron beam is used to scan the surface of structures. Okay, and only the reflected beam is observed. So if you see here, if you have the sample at the end here, and the entire electron beam will focus onto the sample, and the sample will reflect, and the reflected light will be reflected beam will be captured and will be observed. Whereas in case of transmission electron microscope, there you have the sample in between, and the light will transmit, that means it passes through the specimen. So electrons will passes through the specimen. So this basically allow us to see thin sections of specimen, especially if you want to see what is there inside the cells. So transmission electron is a better idea in that case, and also transmission electron microscope has a better resolution compared to scanning electron microscope. This is how a real electron microscope looks like. Okay, this is how it looks like. This entire thing is in the vacuum, and there will be all the structures, that is the source of electron, that is the electron gun, electromagnetic plates, and your specimen. Everything will be here, and people can observe through these lenses over here. What you see here, they can observe the specimen from here, and whatever they're observing they can see on this monitor as well. So this is all about electron microscope and resolution, and I hope you have understood this basic concept. Thank you so much.